All right, so good morning, everyone. Topics that we're going to discuss are the cell cycle and the cell division. Okay, so what happened during the cell cycle? So it is where the sequence of growth and division of a cell happens. Okay. All right, so on your screen is a graph representation of a cell cycle. So 93% of a cell's life is spent in the interface. Okay, so interface, it is where the period of growth and DNA replication happens. And it composed of three phases. We have the G1 phase or the growth 1 phase. So in this phase, this one, in G1 phase, the cell increases in size. Okay, so remember, each new cell needs a complete set of organelles. That's why during G1 phase, the organelles need to double and increase in size. So basically, it is where the cell grows. Next is the S phase or the synthesis phase. So in here, the DNA is replicated because each cell needs a complete and identical set of DNA. So DNA found on the chromosomes, this structure is a chromosome, and we can found here the what you call the DNA, and it is located in nucleus of the cell. So during interphase, specifically in S phase, each chromosome copies itself. So the two sister chromatids, this one, yung kulay purple, dark purple, and the light purple, that is the sister chromatid. So pag pinaghiwalay yan, this one is a chromosome. Pag pinaghiwalay yung dalawa, that's what you call is sister chromatids. Ano? Okay, so the two sister chromatids attached together by a centromere. This one, yung dot or circle structure dun sa center ng ating sister chromatids, ayun ang tinatawag na centromere. Okay, so the sister chromatids attach together by a centromere to form a chromosome. So each chromosome has a matching pair, or it's called the homologous pair. So the last phase in the interphase is called the G2 phase or the growth 2 phase. So it is where the proteins needed for mitosis are produced. So in here, the organelles also doubled and a new cytoplasm forms and all other structures needed for mitosis. Okay, so the last is the M phase or the mitotic phase. So it occurs in series of steps. So the mitosis or the PMAT steps, it is where the nucleus divides, and cytokinesis where cytoplasm divide, forming two cells. So the steps of mitosis ensure that each new cell has the exact number or the same number of the chromosomes as the original. Okay. So let's proceed in mitosis phase. Alright, so in prophase, the chromosomes are visible, and the chromosomes are visible, which is a sister chromatids. This one, yung mga nasa gitna. And the centrioles move to opposite sides of the cell. So this one. And the nuclear membrane, this one, yung nasa labas, that is a nuclear membrane, and it starts to disappear. So the spindle fibers forms this one, yung guhit dyan, it's the spindle fibers. And ano ba? What is the purpose of this spindle fiber? So it is a fibrous structure from the cytoplasm which forms the centriole. So our next step is the metaphase. So in here, the chromosomes line up. As you can see, they are lined up on the equator of the cell, and the spindle fibers attach to the centromere. This is the spindle fiber, and they are attached at the center of this chromosome. Yung sinabi ko sa inyo kanina na nasa center, or yung circle structure na nasa gitna ng chromosome, that is a centromere. Okay? So our next step is the anaphase. So as what you have observed, the spindle fibers pulls the chromatids apart. So it causes the centromeres to divide. So the chromatids move to opposite sides of the cell. Okay, so now there are two identical sets of chromosomes. Okay, so last is the telophase. So when the chromosomes reach the opposite sides of the cell, the spindle fibers break up and the nuclear membrane begins to reform. Ayan, so katulad niyang nasa inyong screen. Okay, so a furrow begins to develop between the two sets of chromosomes. Ayan. The next one is the cytokinesis. So now we are in cytokinesis, and it occurs at the end of the mitosis. As you can see, the organelles are divided. And there is a division of cytoplasm that forms two new daughter cells. So these daughter cells are genetically identical. After cytokinesis, the cell return the interface. So paulit-ulit lang yung process. Okay. Here's a mitosis animation, and it summarizes the prima and the cytokinesis. Okay. Okay. So, in cell, there is also a checkpoint and control point. So there is this regulatory protein called cyclins that control the cell cycle at checkpoints. So in G1 checkpoint, this one, it decides whether or not cell will divide. Here, it checks whether the cell is big enough and has made the proper proteins for the synthesis phase. If not, the cell goes through a resting period, this one, or in the G0 checkpoint until it is ready to divide. While in the S checkpoint, they determine if DNA has been properly replicated. And last is the mitotic checkpoint, wherein S point, determine the ayan. So, while in the mitotic checkpoint, it ensures the chromosome, it ensures that whether the chromosomes are aligned at mitotic plate. Okay, so here's is an example of the cancer results from uncontrolled cell division. So due to the loss of regulation of the cell cycle, the cancer cells more rapidly reproduce than normal cells. So it increases the number of abnormal cells that may form a tumor. So what is the possible cause of this? It can be cells DNA is damaged, and also because of the radiation and the toxic chemicals that enters inside our cell. Okay, so let's move on with one of the types of the cell division, which is meiosis. So meiosis is the process of cell division in which gametes are formed and the number of chromosomes is half, so that the sexual reproduction and zygote formation can occur. So when we say zygote, it is a fertilized egg which has a diploid number of chromosomes. Ayan. All right. So the diploid or the 2N is a cell with two of each kind of chromosome. So one of these chromosome is from each parent. So in order for sexual reproduction to occur, each cell involved must reduce its chromosome number by half. So that is a term haploid or the N, which is a cell with one of each kind of chromosome. So, and so when we say haploid cell, cells, they are the gametes. They can be the sperm cell or the egg cell. So in human, we have 23 haploid cells and 46 diploid cells. So in animal or insects, they have different number of haploid and diploid cells. So we have this computation to get the diploid cell. So the 2N or 2 times the gamete or the specific gamete of an organism. So, for example, 23, 2 open parenthesis, or you will multiply the 23 by 2. So the result of that is 46, which is the diploid cell of a specific organism. Okay. So what is homologous chromosomes? So they are paired chromosomes with genes for the same trait arranged in the same order. So for example, we have your eye color, your hair color, your height, and one may code for blue, blonde, tall. So each homolog may code for brown, blonde, short. So homologous chromosomes may have different alleles on them. When we say allele, it is the gene or the gene form for each variation of a trait of an organism. Okay, so Alright, so this is an structure of the homologous chromosomes. And this one, yung sinasabi ko sa inyo kanina na allele, wherein it is the gene form for each variation of a trait of an organism. Ayan, sister chromatids. Okay. So let's move on with the stages of meiosis. So unlike mitosis, meiosis has two sets of phases. So we have meiosis 1 and meiosis 2. Meiosis always starts in interphase. So where chromosomes replicate, and each chromosome consists of two identical sister chromatids. Ayan. Okay. So in prophase 1, each pair of homologous chromosome come together to form a tetrad. Okay? This one. And what is tetrad? Ano ba yung tetrad? So when we see tetrad, it is a two homologous chromosomes come together and the four chromatids overlap. So what you can see in the illustration, this one, while in Metaphase 1, here, in Metaphase 1, the, excuse me, the homologous chromosomes line up together in pairs. Ayan. As what you can see, the chromosomes are at the equator of the cell. And in anaphase, this one, the spindle fibers attach to the centromere. Attached to the centromeres of each pair. So in anaphase, that's in metaphase. The centromere or the spindle fibers are attached at the centromere of the chromosomes. And in anaphase, here it is illustration, this is the anaphase. In anaphase, the spindle fibers attach to the centromeres of each pair. And the homologous chromosomes separate and move to the opposite ends of the cell. So, centromeres do not split like they do in mitosis. Okay, so now each cell will get one chromosome from each homologous pair. So in telophase 1, the spindle fibers breaks down and the chromosomes uncoil and the cytoplasm also divides. So another cell division is needed because the number of the chromosomes has not been reduced. So after telophase 1, there may be a short interphase, but not always. It is important to note that if a cell does have a second interphase, there is no replication of chromosomes. Always remember that. Okay. So after meiosis 1, we have the meiosis 2. So meiosis 2 is basically like mitosis. So it's the same as meiosis. But remember, the chromosomes did not duplicate in interphase 2. So in prophase 1, in prophase 2 rather, so the chromosomes begin to line up in the middle of the cell. So the spindle fibers begin to form. And in metaphase 2, the chromosomes line up on the metaphase plate. Ayan, metaphase plate. And next is the anaphase 2. So here, the centromeres is split. and the sister chromatids separate and move to opposite sides of the cell. And last one is the telophase 2, wherein the nuclei form and the spindle fibers disappear. So it is where the cytoplasm divides into two. Okay. So the number of chromosomes in each daughter cell has now been reduced by half. This one, as you can see in the illustration. All right. So, and that's for the cell cycle and cell division. And once again, you must comment your name, your section, and the short takeaways about our discussion today. Thanks and God bless everyone. And see you on our next discussion. Thank you.